Okay, hello class. Today we're going to talk about one of my favorite topics, stem cells. Previously in this class we've talked about eukaryotic cells, multicellular organisms, and different cell types in the body. And today we're going to expand on that and move to stem cells. Today during this class we're going to cover three main objectives. We're going to learn what defines a stem cell. We're going to learn the definition and the difference between multipotent and pluripotent stem cells. Next, we're going to cover some different sources of stem cells and types of stem cells. Specifically, we're going to go over sources of pluripotent and multipotent stem cells. And lastly, we're going to go over some real-world applications of stem cells. We're going to cover some of the clinical and research in the laboratory. First, let's move on to what defines a stem cell. So according to the dictionary, the definition of a stem cell is a simple cell in the body that is able to develop into various kinds of cells. So this definition is very limited. It's missing one of the main points of what makes a stem cell. Specifically, a stem cell must also be able to produce more stem cells. See in this cartoon, this is an example of a stem cell. A stem cell goes down one of two paths. It can either replicate and make more stem cells, or it can divide and differentiate into one of many different cell types. Can anybody tell me what differentiate means? No, you don't know.
One cell type to another cell type?
Right. It's when one cell type is programmed to become a different cell type. And that's what's happening with the stem cells here. There are two major categories of stem cells, which you'll hear about. There's pluripotent and multipotent stem cells. Pluripotent stem cells have the ability to differentiate into any cell type in the body, while multipotent stem cells are more limited, whereas they can only differentiate into some types of cells in the body. Next, let's move on to different sources and types of stem cells. Can anybody tell me some examples of stem cells they've heard of, or different sources of stem cells?
Embryonic stem cells.
Right, that's one. That falls under the pluripotent category. Any others?
Bone marrow.
Right, bone marrow. Very good. Is that it? Okay, let's go over what we have in the slides. First, we're going to cover pluripotent stem cells. So again, pluripotent stem cells are the cells that can differentiate into any cell type in the body. There's two main types of pluripotent stem cells currently. There's embryonic stem cells, which Henry just mentioned, and there's also induced pluripotent stem cells. We don't have time to really get into induced pluripotent stem cells in this lecture because it's beyond the content of this course. But briefly, I'll just tell you that they're when adult or somatic cells are reprogrammed to behave like embryonic stem cells. So this cartoon shows how we derive embryonic stem cells. The inner cell mass is collected from the blastocyst, which is an early stage of embryonic development. This is collected and cultured on a tissue culture plate and allowed to expand, to proliferate, and divide into many, reproduce into more embryonic stem cells. And from here, we can induce differentiation and differentiate into many different cell types and tissues. So I know that all of you have some degree of tissue culture experience. Can anybody here tell me what is on the bottom of this tissue culture plate under the embryonic stem cells? Okay. So embryonic stem cells, they're a little bit different than what most cells you're going to encounter in the laboratory. They're very finicky, they're very hard to grow. This is because it's very hard to replicate in vitro and in tissue culture plate their environment in vivo inside the animal. One of the easiest ways to get these cells to grow is not to grow them directly on a tissue culture plate. It's actually to grow them on top of another layer of cells. We call this layer of cells the feeder layer. So this is an example from some research we've done in the laboratory. This is a cluster of human embryonic stem cells. They're growing on top of this layer of feeder cells, which are mouse embryonic fibroblast feeder cells. So these feeder cells help the cells to adhere and to also proliferate, because they are providing many of the cytokines and nutrients needed for these cells to survive and culture. Are there any questions? Okay, next we're going to move on to multipotent cells. So again, multipotent cells means that they can differentiate into some cells, some tissues throughout the body. So there's several different sources throughout the body. Every part of the body has some kind of source of multipotent stem cells. So I'm just going to cover a few of the most important, well, not most important, the most widely studied sources of multipotent cells. The first, which you've all heard about, is bone marrow. So bone marrow has two important stem cell sources that we're aware of right now. The first is hematopoietic, or HSC, stem cells. These are the cells that differentiate into the cells that comprise blood. Another very important stem cell population in the bone marrow are bone marrow stromal or mesenchymal stem cells, or MSCs. These cells are known to differentiate into several tissues and several cell types, including bone, fat, and cartilage. Another very popular source of stem cells in research is from fat, and these are called adipocyte-derived stem cells, or stromal cells. Can anybody tell me why fat is such a popular source of stem cells in research?
Because obesity is a prevalence.
Right, it's just very easy to get. It's very easy to isolate fat cells as opposed to bone marrow or other sources of stem cells. And if you look into literature and research done in laboratories, you can see that there are many types of tissue, different types of cells that these fat stem cells are able to differentiate into. And this is just a short list, there's many more, but some of what's commonly found is differentiation into fat, bone, cartilage, muscle, and endothelial cells. Lastly, another very popular source of progenitor cells, of stem cells, is umbilical cord blood. Umbilical cord and the blood are very rich in stem cells. The blood is very rich in hematopoietic cells, HSCs. And again, these can differentiate into the cells which comprise blood. Are there any questions? Okay, lastly, we're going to cover some real-world applications of stem cells, including what's seen clinically and in research. So right now in the United States, clinically, you're going to see use of bone marrow and umbilical cord blood. And these are primarily used for the HSC, or the hematopoietic stem cell content. These are the stem cells which can differentiate into the cells which comprise blood. And mostly what you're going to see this used for is to treat blood cancers such as leukemia and lymphoma. Under investigation in the United States, there are many clinical trials right now using stem cells beyond bone marrow and umbilical cord blood, and for other applications of bone marrow and umbilical cord blood. Right now you can see that there's trials to treat, using stem cells to treat neurological diseases, central nervous system damage, spinal cord damage, brain damage, cancer. There's one study to use stem cells to help aid bone healing, and many, many more. If you're interested, you can just look under clinicaltrials.gov and search stem cells just to see the wide range uses for stem cells in clinical trials right now. In the laboratory, if you do a literature search on like one of the search engines that we talked about previously in this class, such as PubMed or our Web of Science, you can see that there's also lots of research on stem cells from different sources. There's many different sources of stem cells being researched, and there's also lots of characterization of these stem cells. For example, there's lots of stem cells right now being researched from muscle called satellite cells, which may play a role in healing bone. There's also lots of research with tissue engineering. Specifically, it's found that if you combine stem cells with a biomaterial or like a synthetic scaffold for tissue healing, that you can see that the stem cells can help aid the healing. Can anybody tell me why we're not seeing any clinical research with human embryonic stem cells?
It's not legal.
Yeah, not legal, of course. Any other reason?
Difficult.
Yeah. Okay, there's ethical issues, legal issues. They're not the easiest to harvest. Also, one of the issues are safety issues. So embryonic stem cells, they're pluripotent, which means they can differentiate into any cell type in the body. If you use embryonic stem cells and you use them as is in the body, they can differentiate into anything, not necessarily what you want them to differentiate into. And what they're going to do is develop into something called a teratoma. And that is a tumor that's comprised of different tissue types. It could be anything from the body. So they're very dangerous. It's much easier to work with cells that are either partially differentiated or some kind of stem cell that is already prone to differentiate into some certain lineage, some certain cell or tissue. Okay, I'd like to recap what we learned today. First, can anybody tell me what defines a stem cell?
A cell that could replicate to another stem cell and also differentiate to other cell types.
Right, everybody hear that? A cell that can either replicate itself or it can differentiate into a different cell type. Okay, can anybody tell me an example of one of the sources of stem cells that we learned about today?
Cord blood?
Right. Cord blood, bone marrow, fat. There's many other sources. And can anybody tell me a real world application that we either talked about today or one that I did not mention?
Tissue injury?
Right? Yeah, treatment of leukemia. With bone marrow or cord blood transplants. Anything else? Well, do we have any questions from today's lecture? Any burning questions about stem cells? No? Okay, well, thank you very much. I want to leave you with this thought. I want you to think about stem cell research. Think about what's been done and what's in progress right now. What's going to happen in the future. There's much more on the horizon for stem cells. Okay, thank you.